do roll-to-roll printing for. All the optics, electronics is printed on this flat sheet of paper. You fold it, and you get an instrument for a price point of $1 single that does 700 nanometer resolution imaging. So how many do you all have a sense for 700 nanometer? I'm just trying to quiz you from a context of lens scales. So think of this as take your hair, take a knife, chop it 100 times, and that one slice of that hair would be 1 micron. When you start thinking about lots of bacteria around 1.2 microns, and so it starts to really hit the limit. And one of the contexts of thinking about tools is clearly as you add more complexity to tools, you can really go further. But here, the reason this is interesting is from a perspective of cost. That the fact that you can fundamentally do this at a price point in parts for $1 single. So there's a lot online about this, so I don't want to actually talk about this project, but I'll just mention one thing. From a healthcare perspective, we spend a lot of time. I mean, one of my biggest passions has been to be out in the field, and one of the reasons we invented this tool to begin with was not for citizen scientists per se, but for really healthcare workers to really diagnose diseases, because it turns out healthcare workers are out in remote places where there are no instruments, there are no tools to begin with, and they are doing science that is very important for a person's life. If you get malaria, falciparum, you have 7 days to be either diagnosed precisely yes or no. And that 7 days, at the end of those 7 days, you die. So that's really almost on the edge of starting to think about what scientific tools should be doing and where do they need to go. So that's malaria up there, that's E. coli, bacillus. But then when I started doing things like this, we do field trials in Uganda, Ghana, Nigeria. The other thing started to grow is one of the contexts that we would go to many of these places, we would spend a lot of time with kids in schools as well, because they are the new force that will essentially enable healthcare in any places. And then we started broadly exploring what happens to the world. And this is sort of a vision that we are essentially going for, where every single kid carries a microscope at all times in their pocket. And the way this instrument is designed is really think of a ballpoint pen. You've all carried a ballpoint pen. And that ballpoint pen is a tool that's completely changed. You know, anybody can write and express themselves. So we're trying to do the exact same thing for microscopy. And this is literally not a joke. That is our mission, is thinking about what happens to the world if every single kid carries a microscope in their pocket. And I have a few of these to play with. I can talk a lot more about this, but I'm going to run out of time, and I'll tell you about something else. So the one thing that we are doing before we release the instrument out, openly available, we're doing another science experiment in the middle. We call that a 10,000 microscope project, where the goal is just like a ballpoint pen. If I gave everybody a ballpoint pen, but then nobody learned how to write. There is something conceptual about science that doesn't get talked about that often, which is a very simple idea of learning how to ask questions, building a hypothesis, coming up with something, and then re-asking that question. So we're trying to do something like that with 10,000 people around the world. We just sent out a call. In around 2 weeks, we had 11,000 signups from 130 countries in the world, and the requirement for the sign-up was that they had to propose one experiment that anybody else could replicate. One single experiment. And just, I read these emails, I'm just completely blown away by how science is so contextualized. There's a farmer in Mongolia who's been struggling to convince his farmer friends that camel milk after a certain date does expire. And the fact that they should, and he's discovered this pathogen, that bacteria that grows in it, and he's trying to convince them, but they have never listened to him. There is another farmer, actually, in India who's discovered this fish that was for a pet market, and that fish essentially eats mosquito larvae that's specific to dengue. Dengue is coming up in a big way in India. He wants all the farmers to carry this fish, except to keep the fish healthy, you need ciliates. And so every farmer needs to check on the ciliates' health to really be able to actually transfer. And this is what from my perspective, when I think about citizen science, I, sitting in my own office, would never imagine these applications. There is a Canadian biologist who's sending our microscopes on a nanosatellite, primarily because the weight criteria is so tight. He can afford all the microscopes in the world, but he needs that 10-gram limit. and we fit in that bill. So there is this context of starting to think about openly letting people ask their own questions. This is why I do science. If somebody had to tell me that I have this big goal, I'll discover something if I contribute in this specific narrow way, I might not be as excited about science. For me, science is most important primarily because it's my question, and it doesn't matter whether a German guy in the 1800s already discovered it or not. It's a discovery of one. And so what this project is really trying to do is we're going to curate 10,000 examples, and this acts as a backbone of starting to think about a biology manual that's not written from a perspective of, here is a book, learn all these things from a book, and then you can ask a question. And the physics community has already done it. So if you think about why is the sky blue, think really hard about that. And you'll realize, depending on your level of comfort, you can keep answering that question in a deeper and a deeper layer. So what we're trying to do is do something like that from a biology context. And I can talk a little bit about types of questions that people are trying to poke with something like this, which will become completely available as a platform for microscopy and biology. I'm going to end here and just show you one quick project which is completely unrelated. And this should only take less than 4 minutes or so. Do I have 4 minutes?
Yeah, that's fine.
Okay, so we're going to switch context because I wanted to tell you something about that you might not have heard of, another project in the lab. The lab is kind of split between, I kind of call frugal science and marine biology, and another frugal science project that came out of some of the field work that we do. This is very hard to see, but this is a road that cuts Uganda in half. You can see it's a dirt road. Other than a train, there is literally nothing. It's a forest. You keep going. This is the point in Africa that has the highest malaria incidence from a perspective of the probability of if you were bitten by a mosquito, you're bound to have malaria. And so when I think about contexts like this, and we think about as scientists, how do you really tackle problems like this? We go and work in this field site for a specific reason. I mean, A, because this is a very diverse place, but B, you kind of bump into sites like this that really fuel you in terms of what are we doing. This is a picture that I took accidentally that bothers me a lot, which is a mother with 4 kids of the neighbors, and those 2 little benches that kids are sitting on are graves of their own siblings. And one of the contexts that comes in from a perspective of science is this is a tradition that's done in Ugandan families because eventually you want to remember. And this is something that haunts me back from a perspective of science has gotten us to the moon, but we haven't solved some of the very basic challenges as well. And so one of the contexts for this is really the global context. That's what is an important perspective here. So I'll tell you about one project which is related to engaging a broad range of people in science, but in a very different problem. And so it's a 2-minute video which I'll just narrate. So everybody's read the Bill Gates tweet by now. So think about mosquitoes as one of the most dangerous animals. One of the things that ends up happening, out of that 7 billion population, almost around 50% of that population is in danger of at risk of a whole bunch of diseases, including new viruses that keep getting discovered. They're mostly around the tropical belt. One of the contexts that you start thinking about, you hear malaria quite a lot. We can't even count how many people die every year of malaria. This number gets contentious every single time. One of the things that you have to think about, why is something so simple and it's been bleeding the planet for centuries? You can read Roman history and you'll find examples of this. And why has it been so difficult to actually tackle a simple mosquito? One of the perspectives that we think about this is really actually the spatio-temporal dynamics. You can only control something once you understand in the environment, how does it change? Where does it go? There are 3,500 species of mosquitoes. Many of them have many different hosts. Many of them carry a whole bunch of completely different new parasites to begin with. That are fairly dangerous. There are many of them that we have absolutely no disease drugs for. And one of the contexts that ends up happening is how do you tackle a problem like this? You tackle a problem like this, it's called human baiting. I go out in the field as a researcher, I roll up my sleeves, and I catch a mosquito on my arm, because it has to be a human biting mosquito. You catch tens of thousands of those mosquitoes, because the probability of finding one that's infectious is fairly low, but the reason the disease still spreads is because you have tons and tons and tons of mosquitoes, trillions of mosquitoes. So this is almost like a problem from a scientific perspective of finding needle in a haystack. And one of the things that we started thinking about it is, and this is where people become very important as well, is how do you really spy on mosquitoes? And how do you really think about a real-time map? You know, all of you have read the, actually something came out just yesterday about Aedes aegypti, a dengue mosquito that's been found in Santa Clara. So one of the contexts that a brilliant graduate student in the lab as an experiment did, we started keeping mosquitoes without knowing what we're going to do, and she fed them jello because they were dying. And she discovered that every time when a mosquito bites in a hydrogel, it actually releases a tiny drop of saliva. It's a behavioral assay. And so from that, what we've built is essentially a platform that we call a mosquito surveillance platform. It looks like a little card, has 10,000 to 100,000 little spots that are exactly the size of a mosquito proboscis. Mosquitoes bite on it, and they essentially, this is baited with odorants that humans generate. You get tiny salivary samples of individual mosquitoes. You ship them out to a central place. We do quantitative droplet PCR in those. And the other thing that we discovered is when mosquitoes bite you, they're also injecting their own DNA. So we can detect both the pathogens and the mosquitoes. The vision for something like this, and this is why I'm kind of here pitching this out in this audience, is you're going to go to Walmart and you're going to get a card which you will post outside, and then that would be inoculated. You post it back, we do central processing, and we can truly build a real-time map of these drones that are flying around with what pathogens that they're actually carrying. And one of the contexts that starts to happen in something like this is from a perspective of, now we're talking completely new viruses that nobody has ever seen before. So we have a collaboration in the Amazon. We're trying to look at the edge of the Amazon, why do new viruses come out of that edge? And it turns out that when hosts and many mammals are killed because of deforestation, these mosquitoes adapt to new hosts. And guess who is the next new host? And so this is this fascinating world of diseases that impact people, but there is no way to sample the world. And this is where this context comes into play, where if the tool is cheap enough, so the tool that I just showed you, this is a picture of what it actually looks like currently. This is what we've built, and we've done the field lab trials and semi-field trials, and now we're starting to plan and how are we going to do some of our own field trials before we roll this out. But the chip really looks like this. That's really the number of spots. If you've ever seen microarrays and microspots, it's very similar in analogy. I'm going to stop there, and I was going to show you some data, but it doesn't matter. It's cheap enough for us to do, and one of the contexts is, this is a slightly different type of a project, which is really kind of designed around the environment and sampling the environment in a very different way. So I'll stop here, and we'll take questions later.